Yes.
Okay, good afternoon, everyone. And thank you all for coming to the 2025 National Academy of Science Lounsbery Lecture. The Richard Lounsbery Award is given by the National Academy of Sciences and the French Academy of Sciences to recognize extraordinary scientific achievement in biology and medicine. In addition to honoring scientific excellence, the award is intended to stimulate research and encourage reciprocal scientific exchanges between the United States and France. I'm very excited to introduce today's speaker, Dr. J. Shendure, who is the 2019 recipient of the Richard Lounsbery Award. Dr. Shendure is a professor of genome sciences at the University of Washington and Howard Hughes Medical Institute investigator. Dr. Shendure completed his MD PhD training at Harvard Medical School in the lab of Dr. George Church, where he was one of the first to show a proof of concept for next generation sequencing. In his lab, they have established many new techniques and insights into developmental biology, including pioneering exome sequencing, generating massive data sets that enable precise insights into cell fate decisions, and developing gastruloids to understand developmental signaling, to name a few. He is the recipient of numerous awards and honors, a small subset of which include a 2013 NIH Director's Pioneer Award, the 2021 Kurt Stern Award, and the 2022 Mendel Lectureship. It's been fantastic to have Dr. Shendure at Stanford today to share his wisdom and engage in conversation with colleagues and trainees. I can say personally that Dr. Shendure's work is inspiring to the point where even last night, I was up at 11 p.m. with another graduate student pouring through the data his lab has generated to inform new ideas. With that, please join me in welcoming Dr. Shendure as we have the privilege to hear about new ideas in his lab at this year's National Academy of Science Lounsbery Lecture.
Thank you, Arjun, for the introduction, for hosting me today. And thanks all for coming here. This was actually supposed to happen in March 2020. So it took us a little while to reschedule, but we got there. Okay, so my lab is broadly focused on really this interface of genetics and genomics for over 15 years now at the University of Washington. Interface of technology development with genetics and genomics. In the last couple of years, this is primarily what I'll focus on today. We've really turned our focus to a new area. And I think the best way of framing this is just thinking about kind of what really limits the progress of genetics. So I'm of the Sidney Brenner School, which argues that technology is the rate limiting factor in most things in science, right? But even within that, you could argue that within genetics, we were previously arguably limited by our ability to genotype, right? And that, if you look at the past 50 years, it's really been an evolution of genotyping technologies, starting with RFLPs and progressing up to whole genome sequencing for now extraordinarily low costs, right? And so that was a prior area in which we spent a lot of time. Another area in which we and others have spent a lot of time is really kind of modalities for thinking about how we do genetics, right? So how do we connect phenotype and phenotype? What are the paradigms or frameworks that we engage with? And the classic ones being linkage or QTL mapping, and progressing in more recent years to things like exome sequencing and GWAS. But I would argue, looking forward, the place where there's the most room for growth from a technology perspective is how we phenotype, right? So the ways which we phenotype are arguably still quite crude. Okay, and so with that, that's kind of the lens through which we're this most recent evolution of our lab, which has been to really dive into biological development of the mammal as kind of a motivating framework for tech dev. And the genome is fundamentally a program of development. That's really what it is. This could be viewed through the lens of, a single human or differences between humans. It can also be viewed through the lens of, different genomes, essentially encoding blueprints for different programs of organismal development throughout the kingdom of life, and what Darwin called endless forms most beautiful. Yet, we're now 40 years on from the seminal work from John Sulston, over 40 years, and the worm remains the only major model organism for which we have something approaching, or actually in this case, actually achieving a comprehensive landscape of what this program of development that the genome encodes looks like, right? And he did this the hard way, sitting at a microscope for 2 or 3 years, recording things by pen and paper with Nomarski microscopy, which had recently become available. And I think was able to leverage a few features of C. elegans to make this slide. So one is the fact that it's, of course, transparent. The lineage is invariant, and so you can piece together observations made across visual inspection of many different animals, and then, of course, as a relatively small scale. As we turn to something like the mouse, we have these things all look much more daunting, right? It's opaque. Development's opaque. Even in a genetically identical background, it doesn't happen the same way every time. And of course, there's the question of scale. So, nonetheless, I would argue this is still a very well-motivated problem, right? You can think about, again, this program of development as being the phenotypic equivalent of the Human Genome Project. And I think we should be unapologetic and, sort of defending all model organisms, including but not limited to the mouse, as terrific models for deeply exploring human biology. Okay, so now zooming way out, right, to think, just on kind of a first level paradigm level, kind of framing, how do we actually measure biology over time, right? And there's really only a handful of options. The first is to observe, through microscopy or similar techniques, how something unfolds over time. And this option is not as I mentioned before, not really an option as it was for the worm for the mouse, because of the opacity of mammalian development. Our other option, or our second option here, which I'll delve into, and I'll come back to the third option, is to sample. And this is mostly what we do in omics, right? Where if I want to measure a time course, I simply destructively take a lot of samples of whether it's in the cell culture hood or in vivo, and I break those cells open and make my measurements. And then I even though these are derived from different samples, I try to piece together what happened over time, right? Nonetheless, if there's some consistent process there. So this is one tack that we've taken for mouse development. And so this is not to scale, right? This is going from a gastrula to a mouse pup a few weeks later is a roughly 3,000-fold expansion in the number of cells. And this unfolds very quickly. But what you can imagine trying to take a series of snapshots and then trying to piece together what happened along the way, right? And so there's obviously a lot of cells here, right? This question of scale. In our lab, along with Cole Trapnell's group over the last, roughly nearly a decade now, I guess actually a decade now, we've been investing in one particular paradigm for single cell molecular profiling, which is this idea of a split pool framework for combinatorially barcoding cells or nuclei, such that one can obtain barcodes associated with the nucleic acids that allow you to deconvolute which cells derive from the same, which reads derive from the same cell or nucleus, and thereby profile large numbers of cells without ever actually isolating each cell in a single volume, whether that volume is a droplet or a well. And so just our two labs together over these last decade have been able to profile around 50 million cells. And I think the field more generally, the workflow lends itself to adaptation to a wide range of phenomena that one might want to profile at single cell resolution if you can find a way to couple the barcodes to that phenomenon of interest. So this has been a really fun journey. In the context of the mouse, and I'm going to only briefly talk about this approach because I want to save time for other things. So I'm going to fast forward to like our most recent crack at this this sampling paradigm, taking credit for mouse development. But I think it also is a nice illustration, I think, of how far the field has come, both at the experimental level and the computational level. I think this is the kind of project that might have once been the provenance of a genome center, right? But it was really just 3 people who did all of the work, essentially. There was one, a collaborator at Jackson Labs, Ian Welsh, who heroically profiled like 500 mouse embryos, kind of morphologically staging them and selecting around 75 for us to go forward with single cell RNA-seq and essentially spacing them at 2 to 6 hour resolution. And then with these combinatorial methods, Beth Martin, who's a research scientist in my group, was able to do all of the experiments in a month or two, profiling over 10 million cells that are from all these embryos. And then finally, one graduate student, now a postdoc, Chengshan Qiu, was able to do this unified analysis of all the data, right? So it's really a testament, I think, to the multiplex methods, but also I think the free availability of bioinformatics packages that are well developed and widely useful and can scale. Okay, so I'm just going to give a few mini anecdotes from this work before moving on to other things, but just at the highest level, to give you a feeling for how we piece together and summarize what happened over, again, it's only 2 weeks from E8 to birth, but an incredible amount happens over that window. And so really this comes down to a task of trying to connect cell types to other cell types, or cell states to other cell states, I should say. So one point of reference note here, and I'll come back to this towards the end, is when we generate these kinds of data sets on densely sampled processes, we don't necessarily end up with a bunch of clusters, but rather we end up with more of a continuum, right? At the extreme, what you're going for is a tree, right? Much like the tree of development. So here we have 4 cell types that we've classified with different labels, but they're not discontiguous from one another, right? And so if we project them together in a UMAP, we can see this continuum, and it really boils down to finding these interfaces where there are mutual nearest neighbors that traverse, some boundary of calls or labels of different cell types, and then simply creating a directed graph, right? We can, if you just looked at this boundary, you would say that these 2 things are connected, but by seeing that which cells are older and which cells are younger, we can orient the graph. And we do this over and over, and this is not a small amount of work. Me and CX spent about 2 years, largely during the pandemic, just staring at UMAPs and trying to, so it's a heuristic approach to put this all together, but essentially we came up with kind of a V0.1 roadmap of cell types, leveraging our data, which starts around late gastrulation all the way out to birth, but by also piecing it together with other data sets, such as a Pijuan Sala data set generated by John Marioni and Wolf Reek, and other data, we can actually go all the way back to the single cell zygote, right? And so this is kind of a framework for where we eventually want to get. So, but really briefly, a question I often get, like, did you find anything new? And this whole process is one where you find, yes, we found a lot of things that were new, but then we go in the literature, and somewhere buried in some Dev Bio paper from 1997, there's some reference to the phenomenon that we're seeing, right? So I think about, a lot of what we found is actually not new, but I do think that there's a value here nonetheless, even if we discovered nothing new about the systematization of knowledge into a cohesive framework, right? On one hand, you have the robustness and uniformity of mouse development, which has been tackled as a million puzzle pieces by different groups, but ultimately this is one process that we want to define and describe in a unified way. And also the systemization of nomenclature, which is also something that I think is the direction the field could benefit from advancing on, right? But there are certainly errors in here and a lot of reductionism, so to speak, right? But I want to mention one thing that was new, at least, right? Or at least new to us. And so this is a snapshot of data from proximal tubule, actually the whole kidney, right? So this I apologize, there aren't cell type labels here, but essentially these are nephron progenitors that are going out in kind of distal or proximal directions to give rise to functional nephrons. Here they're colored by cell type labels. Here they're colored by time points, and you can see the early time points here differentiating out in both directions. I think you can appreciate something, which is that this is really a continuum. You're not seeing one color seamlessly blending into the next. And I think, at least the way I think about this in my head, is that you imagine cells rolling down a Waddington's landscape. As development proceeds, they kind of get desynchronized, right? And so you have very similar cells that are from a diversity of time points within some constraints, right? And this unfolds as long as nephrons continue to be produced. And so one exception to that kind of process of, that continuity, if you look quite carefully, is right here, which is that the P0 proximal tubule cells are quite distinct from pre-birth proximal tubule cells, right? And so this was processed, a slightly different batch. And so the first thing you worry about is like, this is batch effects or something like that. And then you look at other cell types, because it's kind of weird if we only saw it in the kidney. Oh yeah, so I should have said here, these are P0, these are earlier than P0. And we see it in other cell types, right? So in contrast with the bulk of the data set, where we see really a continuum, there are these discontinuities between mice from before E17.5 versus, or up to E17.5 versus birth, where the data just kind of leaps, right? And so, you think to kind of hopefully rule out batch effects, we can ask whether this replicates in a more careful experiment. So now, rather than just kind of waiting for the mouse to be born when you show up in the mouse room the next day, Ian tried to stick around and do a more controlled C-section type thing from a single litter, and in particular harvesting mice. He's going to have some birth naturally in this litter, and then some that he's sacrificing after certain numbers of minutes, right? He went to lunch, right, which is understandable. People need to eat, but he came back and the first three mice were born, so he thought quickly and harvested, but we don't actually know exactly how old these three are. But the rest we do, at least, relative to the time of C-section. And here we see a similar phenomenon. We can narrow it down. This is essentially a UMAP of cells colored by the time point of the C-sections. And you can see this very rapid transition around 40 to 60 minutes in each of the cell types in which this phenomenon is the most prominent, right? And so what's going on here? It may be obvious to many of you. For whatever reason, it never occurred to us. But this mouse is born, right? And if you really step back and think about it, if you've ever been in a labor and delivery room, this is by far the largest change in circumstances you will experience in your life, right? Apart from perhaps death or maybe fertilization, I don't know. Where you're, this change, where this transfer to from an intrauterine into an extrauterine environment, right? And so perhaps it's not surprising that we see these very rapid changes, right? In particular, and I should say that we're seeing huge numbers of genes changing. So this is really just looking at what are the key patterns that stand out, but there's a lot more that appears to be going on here. It kind of fits with what you might expect for these kinds of transitions, right? So you no longer have access to mom's sugar, so you need to turn on gluconeogenesis. The world is cold, right? So you've got essentially uncoupled respiration turning on in brown fat. And then, here the genes kind of make sense. In the lung, it's entirely obvious why things would need to change up, right? You're breathing. But the genes underlying that don't necessarily form a cohesive story. So that's something we still haven't really fully puzzled out, even though there are clearly enormous changes going on. And then these are the most prominent examples, but really in every cell type, except for neurons, we see this phenomena of rapid change around birth, right? And then just one more little nugget here, which is that there are also pretty significant differences between the pups that were birthed naturally and the ones that were born by C-section. And there's speculation about how differences in modality of birth might have long-term outcome impacts. And so this is kind of an interesting model to think about, and one that we're delving into further now. Okay, so punchline, right? The Lewis Wolport's the Dev Bio crowd, I think, right? Gastrulation, and I'm not disputing this, but maybe birth is number two. So, okay, so cell types and cell lineages are not the same thing. So I started with the example of Selston, but then I went into this map of cell types, which is very far from actually tracking cell divisions, right? This is like the Cliff Notes, right? And what we want is the actual extremely detailed description of every moment that happened in a particular day, so to speak, right? Okay, so this brings us to the third paradigm of recording, right? Okay, so the big goal here, or the big idea, is this question of whether we can turn cells and organisms into their own historians, essentially leveraging the machinery of the cell, including the genome, as both a substrate for encoding the functionalities of a typewriter, as well as the substrate for writing to that typewriter, not to mention ribosomes, et cetera, et cetera. And to do this in every cell throughout development over time, right? So just to put some context on how long I've been working on this, this was my first project in graduate school. In my first 6 months in the church lab, was the idea here with Sue DeMecki. And we had this broad concept. This was before Brainbow and things like that. We were going to build this big bit flipper and put it into a mouse. And all that came out of it was a patent. I gave up after 6 months and moved on to other things, which I think was unquestionably the right decision at the time. But the idea kind of stuck with me. And years later, when I had the opportunity to put my own graduate students on it, I had my own student flail on this for a few years, Aaron McKenna, before he finally saw the light, which was a different path than we originally conceived of when CRISPR tools came into wide use, right? And so he collaborated with Greg Finley and Jamie Gagnon, who was a postdoc with Alex Shear, on this idea called this concept of essentially having a transgenic barcode of target sites that we then allow CRISPR to stochastically introduce small insertions and deletions mediated by non-homologous end joining to this barcode over developmental time. These are irreversible, they're cumulative, and they just kind of happen along the way, but in ways that are different in every lineage, because NHEJ may result in different kinds of indels in different subsets of this lineage. And so the kinds of patterns we get looks like this. This is data where every read here is the transgenic barcode in a different cell. The red corresponds to deletions and the blue to insertions. And you can clearly see patterns here, right, of commonality and difference. And this can essentially be the fodder for phylogenetic reconstruction, much as we reconstruct lineage trees of SARS-CoV-2 or species or anything else where there is some process of mutation, right? And so this was quite some years ago now, and many groups had similar ideas around the same time. There's been terrific work, I think, from Alex, Sarah Bowling, who's here, many other groups, Jonathan Weissman, kind of taking this paradigm and pushing it forward. But I think there are limitations, right, that we encountered over time. One is that the editing events are unordered, right? You can see, this is our barcode of 10 sites. You can see these large deletions. So you've got the loss of information and the kind of death of your recording substrate over relatively short windows, and other limitations that I'll come back to later. But this kind of sparked us to think about whether there are other approaches that build on Gestalt, the Gestalt concept, but try to kind of take this to the next level. And so Junong Choi, who was a postdoc in the lab a few years ago, now has his own lab at Memorial Sloan Kettering, came up with this idea of whether we could record information over time in kind of an ordered way, quite analogous to how a typewriter works, where you're writing left to right, but in this case we're writing 5' to 3', right? And moreover, we're writing symbolic information, right, to the genome. And this is a good analogy, I think, because it really, as you'll see in a few minutes, it really does work in many ways like a typewriter. We've got a single type head for a given tape, and we're punching information into the genome. And what we punch into the genome can be any one of a number of symbols, right? So devil's in the details, right? So here's a little more granularity on how this works. This is a repeating, take my word for it, it's a repeating 14 base pair monomer. And this is our tape. And we structured this such that the first instance of this monomer plus the first half of the next monomer plus this key sequence, which precedes it, collectively form the target site for a guide RNA. And in particular, a prime editing guide RNA. So we're relying on David Liu's prime editing system, which allows us to encode both the site to be edited and the nature of the edit in the same molecule, the PEG RNA. And so here, when we make an edit to this site, it writes an insertion. So everything we'll talk about will be insertions. And that insertion includes two components. First, a barcode, which is the symbolic information that we're writing into the tape. And then also this 3 base pair key, which is the same as the key we had here, which effectively shifts the position of the type head or write head down by 1 unit along the tape. Right? So this is like a processive process, punch, move, punch, move. And the reason that this site and this site are not active at the outset is because they lack that key sequence. Right? Okay. And then the number of symbols that we have conceivably to be able to write is going to be 4 to the x, where x is the length of this barcode. So with a 2 base pair barcode, we can write 16 different symbols. With a 5 base pair barcode, we could in principle write 1024, right? Okay, so one of these things where you don't know if it's going to work, you have the idea, you try it, and it just works out of the box, right? So Choi was able to show that if we have this tape set up like this, just a 3-unit tape, the red box indicates where the type head is, and we introduce 3 different PEG RNAs, each writing a different dinucleotide symbol, we overwhelmingly get directional patterns. The patterns that we get back are left to right, or 5' to 3'. The second site doesn't get written to unless the first site has already been written to, for the most part, right? Okay, so now can we use this to record cell lineage? So in this experiment, we're starting out with a single monoclonal HEC293 T cell, in which we've introduced the prime editor. Many of these, of these kinds of tapes, right? So many instances of them throughout the genome. And we also have many PEG RNAs in the system that are writing different symbols to that tape. We then expand that single cell out to roughly 1 million cells, and we can do single cell RNA-seq to recover transcriptomes, but as part of that transcriptome, to recover these tapes from each of many single cells. And so when we do this, this is just a highly selected subset of 24 cells to give you a sense of the patterns, right? So here I'm showing roughly a dozen different tapes that are each 6 units in length. And you can see that if you look at any one of these instances, we're writing 5' to 3'. We've got some blank tapes at the end here, but generally you don't see tapes written towards the end unless the preceding tapes have already been written to. And the colors correspond to different, in this case, hexanucleotide insertions that have been written to the tapes, right? And you can see these 24 cells happen to fall into 2 clades of roughly a dozen each. And you can see patterns that exclusively occur in this clade versus this clade. And that's what allows us to separate these with some pretty basic phylogenetic tree building. Okay, so now we're only limited by the number of cells we can profile, right? Which can be substantial. And so we can build these very large trees, right? So this is much larger than the C. elegans tree, about 10 times larger, but it's also far more boring. Probably the most boring tree ever built, because these are all HEC293 T cells, right? And are largely transcriptionally indistinguishable, right? Okay, so what do we have? We have longitudinal recording of cell lineage in HEC293 T cells. I'm trying not to oversell this at this stage, right? So we've got our cells, we've got symbols, we've got a lineage tree, right? But kind of a boring one. And we have the symbols being stochastically written to the tape, right? Okay, so now for the second part, I'm going to talk about how we might introduce this to more complicated systems, such as those that start from stem cells, right? So I started, you saw this slide earlier, endless forms, most beautiful, right? We are increasingly in a world where we can recapitulate amazing form and function in the lab in the form of these kind of stembryos, right? I would similarly characterize this as endless forms, most beautiful. One of the dirty secrets that often gets swept under the rug a little bit, right, is the fact that many of these protocols can be prone to irreproducibility, right? And I mean that not only in the sense of, lab A tries to implement lab B's protocol and can't get quite the same results, but also in the sense of intra-batch heterogeneity. So I've got one batch of organoids and they all look a little bit different, right? This is not uniformly true. There are some protocols that are more robust than others. And I think this is both annoying, but it's also interesting, right? Where does this heterogeneity come from, right? Okay, so we've in particular been focused on gastruloids as one model for kind of testing out some of these recording systems in the lab, and for other reasons. So these are a powerful model of early development pioneered by the Arias lab some years ago. They're gastruloid. They do model aspects of gastrulation, but I think of them as primarily an in vitro model of the posterior embryo, where you're applying a Wnt agonist to an aggregate of stem cells, whether mouse or human, and you're getting symmetry breaking, primitive streak formation, tail bud formation, and really what most people are interested in when they study this is the NMPs bifurcate, coming to be and then bifurcating into either neural, posterior or neural versus paraxial mesodermal lineages, like somites, right? And so this is not our data. This is images from Prisca Liberali's group, and well as measurements of cell type composition of individual gastruloids from John Marioni's group. But you can see the heterogeneity here, right? So every gastruloid is a wee bit different, right? Okay, so what is the origin here, right? You can think of a lot of possibilities, right? It could be something environmental, like just differences from well to well. It could be stochastic effects. But one of the challenges here is that, generally when, with a handful of exceptions, generally when people make organoids, they're not starting with a single cell, right? You're starting with an aggregate of iPSCs or embryonic stem cells, right? A few hundred to a few thousand, right? And so the heterogeneity of what stem cells happen to be in that aggregate is one source, potential contributor to the heterogeneity that ultimately emerges when we get our quote polyclonal gastruloid, right? So as one line of attack here, but also because of our interest in trying to model recording in systems that start with a single cell, Sam Regalado, who's an MSDP student in the group, developed a monoclonal protocol for generating gastruloids, right? So, welcome. Here, we're starting from a single stem cell. We grow this up to an aggregate, right? So we're still starting from an aggregate in a sense, right? In the sense that when we do the application of Chiron, we have that aggregate. But now the cells in this aggregate are very closely related to one another, right? They're only a handful of generations separated from one another. And these can grow up and elongate and look like conventional gastruloids. Okay, so now we want to do this at scale, right? So we have many monoclonal gastruloids and many wells. We're interested in the transcriptomes and the cell type compositions of each of those gastruloids. So in order to be able to access those, we can do single cell RNA-seq, but we can also essentially barcode the founding cells, the founder of each of these monoclonal gastruloids. And then based on the barcodes that we see in the debris that is left behind from cell death in these processes, we can essentially match our single cell RNA-seq profiles back to individual wells and therefore back to individual gastruloids, right? So if we look at this at the bulk level, so now looking at essentially cells from all of these gastruloids in one UMAP, one interesting thing is that we see actually more cell type diversity than we saw with polyclonal gastruloids. So this is a bit of a surprise. We thought that maybe switching to monoclonal gastruloids might help mitigate some of that heterogeneity, and that was not the case. So in particular, we have some cells that look more like the hindbrain neurons than the typical kinds of things you see with NMPs, which look more like spinal cord neurons, and neural progenitors, not neurons. But then on top of that, we also see differentiating neurons, which is not something we typically see in conventional polyclonal gastruloids. So by virtue of this debris-seq approach, we can break this down to the level of each of these individual monoclonal gastruloids. And whether we look at it at the level of pseudo-bulk transcriptomes of gastruloids, or the mapping back of these profiles onto the pictures of those gastruloids that we took before we started our single cell sampling, we essentially see 4 patterns. So the middle 2 columns here correspond to what we might expect to see with conventional polyclonal gastruloids, where we have elongation, but they, based on their cell type composition, can't really tell from the images here, they're either biased towards somatic lineages or towards NMP-like neural progenitors, right? But in addition to that, we see that some of these gastruloids essentially fail to launch. I really shouldn't call them even gastruloids. They just are essentially like collections of things that look like our starting stem cells, or they fail to elongate and are largely comprised of cells that look like, are neuronal progenitor-like, but look more hindbrainish than spinal cordish, if that makes sense. Okay, so that's interesting. And so we try to take a different tack on this, which was to ask the question, not of what is the origin, but when is the origin, right? And so this is where the recording technologies come into play. So now, very much a similar setup to what we had with the HEC293 T cells, except now we've got an inducible PE max, so it's quite efficient. We again have many instances of our tape. We have many PEG RNAs, all going into a single monoclonal embryonic stem cell founder at a high multiplicity of infection, right? So many copies of each of these. And then with our founding clone, we can basically make founder cells, and we don't start recording until we add docs, which activates the PE max. And so then we're recording the lineage of this gastruloid, or that gastruloid, or that gastruloid, right? And so we get data much like I showed you before with the HEC293 T cell. So this is all data from one, actually, I shouldn't say from one gastruloid, it's actually from 2 gastruloids. And here we got roughly a dozen tapes I'm showing here. We actually have around 70 or 80 tapes like this from many more tapes than we had before. We're writing left to right, and we can build these phylogenetic clades. We are doing single cell RNA-seq to capture these tapes, but that also means that we know the cell type of every cell at the end. And because we have the debris-seq information, we know what gastruloid the cell came from. And in this case, we nicely see that the phylogeny separates the 2 gastruloids. So that's at least reassuring that something is working. So looking at this another way, these are 2 different gastruloids that derive from the same founder of founders. So like a starting stem cell with a particular constellation of tapes and guides. But really turning on the recording here, right? And we build this nice tree. We get reasonable bootstrap values, which is a good measure of how good your tree is. And if you're looking at the data here, and again, CX, who is really driving the analysis here, you can see a few things. So one is, in general, the cell types are quite mixed, right? Which would imply that a lot of the specification is happening relatively late, as one might expect. On the other hand, we can see some clear differences, like just looking at the colors. Don't worry about what they are yet, but you can see differences between this gastruloid and this gastruloid, and overall cell type composition are quite evident. And then moreover, you do see some blocks, right? Where you've got like a coherent clade, so to speak. And I'll get on a little more granularity on this in the next few slides. So here's one example. So this is a single gastruloid, so one well. Here I'm showing its whole tree, and here I'm blowing up one particular wedge. And you can see that there's a particular cell division in which we have a relatively high bootstrap confidence, in which there's a diversity of cell types up here that correspond to the kinds of things that you might expect to see in a gastruloid. But then all of the descendants of this daughter are extraembryonic endoderm, which is not a cell type we expect to see in gastruloids. And I actually struggled a little bit to interpret this. Kyle, visiting UW, and nicely pointed us to a paper where I think there can be spontaneous differentiation of ESCs into this EXE endoderm-like cell state, and that's likely what we're seeing here, right? And I would just note here, and I'll come back to this, this is the 4th cell division down from the founder. Another example here This is a different gastruloid, again, just looking at a subset of the tree, a single cell division in which we're now looking at mesodermal versus neural lineages. And you can see this very gross difference. It's quite obvious to the eye, also statistically, between the distribution of cell types that this daughter gives rise to versus this daughter. And this is at the third cell division, right? Here's another one where it's a different gastruloid, where now we're looking at a cell division where one daughter gives rise to the kinds of cell types, again, you'd expect to see in a gastruloid, and the other, not exclusively, but largely remain stem cell-like, right? And this is now the first cell division of the gastruloid, right? And I should point, I characterize the first one back here as fate restriction, because it's really exclusively EXE endoderm. These are more clearly fate bias, right? Where it's, you're still seeing a diversity of cell type, even if there is a bias there. Okay, so this kind of takes us back a little bit, because each of those examples I showed you, and there are other ones, are all in these early cell divisions, right? So this is before there's been any chiron applied, right? And so this is, this still doesn't completely narrow things down, because, on one hand, you can imagine this is kind of our starting hypothesis, and I think this is still something we believe is going on, is that there are cell intrinsic fluctuations in the states of stem cells that might be biasing the distribution of cell types that they will go on to give rise to many cell divisions later, right? So we're separated here, as if this is the first or second cell division, we're separated by 10 or more cell divisions from the point at which differentiation will actually occur. Another possibility, which is not mutually exclusive, and is actually supported from work from Jared Tochter and Michelle Chan, as well as from Prisca Liberali, is that there may also be spatial substructure here in this aggregate that is the consequence of spontaneous fluctuations in signaling pathways that are then kind of hard coding in where those cells go in the aggregate, and then interacts with the Wnt signal and results in different patterns of differentiation. So likely things are both going on here, but at least we're narrowing down when this occurred, when the roots of this heterogeneity are taking shape to the early cell divisions, right? Okay, so we can push this a little bit further. Uh-oh. Okay. We can push this a little bit further. So let's try and remove, as I mentioned, the experiments I showed you so far, we're really looking at the emergence of heterogeneity after a gastruloid has been founded by that single seeding cell, right? So now let's think about a slightly different experiment in which we start with a single founder, but this is now a founder of founders. And we're going to start recording here, right? And this will, I feel like there's some biotech joke to be made there that I still need to articulate, but okay. Founder of founders, by the name of VC fund that's down the street. This gives rise to an aggregate, right? And then we split these out to a bunch of single cells. And then these go on to found individual gastruloids. And now we've kind of removed any spatial information here. And we're asking whether the lineage relationships of these founders somehow inform or relate to the heterogeneity that we see down here, right? Okay, so this is the first epoch tree, right? And we can take pictures of our gastruloids here, and we can base such a tree on debris-seq as one example. But then, again, we're recording the whole way through, and so, the answer here, just to cut to the punchline, we're still kind of looking at this, but the answer is yes, right? So we still see relationships between, or we essentially see correlations between the phenotypes of gastruloids and their lineage relationships during the first epoch. The relationships are decidedly weaker than what I was showing you before, but they are there. And in particular, with respect to aspects of the phenotype, like the proportion of cells that will remain stem cell-like, as well as the proportion of cells that will have this hindbrain-like phenotype, right? And so that implies that there is some aspect of cell state that is somehow, fluctuating, but also being transmitted in a way that may bias the gastruloids that ultimately come out of this. Okay, and then the other fun thing here was that, essentially we have this is a tree of trees, right, from our founder of founders. And so this is one monophyletic lineage tree of around 50,000 cells from 96 including 96 monoclonal gastruloids. So the outside ring here are the cell types of individual cells. And then, in the inside ring is like these arbitrary clades that we divided things into. But the middle ring are the individual gastruloids, right, that were separately seeded to different wells and give rise to these subclades, right? So, we were able to explore this question about where heterogeneity arises, but it was also, I think, a validation that the kinds of systems that we built in HEC293 T cells also work in stem cells, and stembrios derive from stem cells, which is not always a guarantee, right? And in addition, we have much more interesting information down here at the tips of the tree in terms of cell types, right? Okay, so now so that's good, but, right, this is still like a monkey at a typewriter, right? So now it's like a monkey at a typewriter in a stem cell instead of in a HEC293 T cell, but it's still a monkey in the sense that we're just punching keys on the keyboard, right? And that's the reason that the only thing we're learning anything about is lineage, right? And to kind of put the limitation, frame the limitation a slightly different way, I think the field, us included, have been focused on using these kinds of CRISPR methods to build lineage trees and then layering on single cell RNA-seq to know the cell state identities at the end. But this has limited value because we don't know anything that happened along the way, right? We can guess who this was. We can guess why a particular bifurcation happened, but it doesn't actually tell us like what the event was that made it go this way instead of that way, right? And all of that rich information that's likely there that Sulston could see because he could watch it the whole time. Okay, so now this last part here, how do we assign biological meaning to specific keystrokes on our typewriter, right? And so, from decades of work, there are thankfully terrific transcriptional reporters for a variety of phenomenon that we might be interested in. So, signaling, either developmental or immune signaling or other signaling, cell states, we've got great enhancers in theory, even neuronal firing in principle can lead to calcium influx, which can lead to transcriptional changes. These by and large operate through Pol2 promoters, but essentially you're getting signal-specific transcription. Guide RNAs, as they're typically deployed in mammalian systems, rely on Pol3 promoters. And so the switch here came, Will Chen, working with Choi, developed a system called ENGRAM that essentially allows us to use a Pol2 promoter or a Pol2 setup, in this case, a signal-specific enhancer coupled to a minimal promoter to drive the production of a signal-specific insertion to the tape, right? So here we've got the signal-specific enhancer driving the production of an endoribonuclease, CSY4, which clips at these hairpins, releasing the PEG RNA, which then goes and writes to the tape, right? And so we can show this works for some basic signals. So these are dose response curves where the x-axis is the concentration of an agonist and the y-axis is not expression, but is recording to the tape. And you can see, nice responses there. And I think a key point here is that the insertions that are driven by a particular NGRAM unit are signal specific, right? So I mentioned before we could have 16 or 1,024 of these. So it's particular insertions that are coupled to particular signals. And so this is very multiplexable in principle, right? But here we can actually show this with 3 signals, 3 NGRAM recorders, all in the same cell line system where different patterns of agonists give rise to different symbols being written to the tape. And you expect these 3 to be orthogonal. They in fact are orthogonal, as you'd expect. but it kind of nicely shows at least we can multiplex this. Okay, so now can we combine the two things? So we want to record signal-specific symbols. We also want to record them in order over time. So this is a very basic proof of concept experiment to just showcase that it's possible, that Will did. So here we just got two different NGRAM recorders, one responsive to docs, the other to Wnt signaling. They're both in the same system, geared up to write to the same tape. And you might expect to have two different patterns depending on which signal came first, right? And in fact, you do, right? So depending on what order you go in, you get very different ratios of this pattern versus this pattern, right? As you might expect. A key point here is this is not in single cells. These are ensembles of cells, right? They're getting exposed to the same pattern, and different durations, different overlaps. And so this is still very early, but I think conceptually, at least, we've shown that we can have a variety of different patterns with just these two signals. And at least in ensembles of cells, which have many more tapes than we currently fit in one cell, we can distinguish between these patterns. So there's still a lot of work, to try and cram so much tape into one cell that you could actually get the signaling history of that one cell, but also so that we can not just do a PCA and see different patterns, but actually de novo infer what happened directly from the patterns. But it's at least a start, right? Okay, so now this is our last ingredient here, right, which is the idea of writing biological signal-specific symbols to the same tape, and then hopefully ending up with what, I think Mike Elowitz coined this in the context of their systems, a decorated tree, right? So the lineage tree, covered with Christmas ornaments that tell you, what happened in particular places, so to speak, right? Or particular times. And then the long-term vision here that we hope to aim for is this idea of essentially turning an iPSC line or a mouse into a recording device, right? So the ones I've checked here are things that we already have proof of concept for, but you can imagine that there's all kinds of other things that one might be interested in recording over time, that if you can find a way to couple it to signal specific or interaction specific or state specific production or activity of a prime editing guide RNA, it could essentially be slotted into the same system, right? That's the hope and the dream. But I think one thing we have going for us is, I think it's pretty clear that the mammalian genome, has fragility in some ways, but it also has immense capacity to host orthogonal machinery, right? And so, or orthogonal sequence at least, right? So this is evidence, for example, that the fact that you can put an entire arm of human chromosome 21 into a mouse as a model for Down syndrome, and the mouse, has some phenotypes, but it's largely fine, right? Despite tens of megabases of additional sequence, which is far in excess of what we actually need to do something like this. And so that's where we're headed, just to kind of wrap up, and amazingly stayed on time. Again, this is kind of the goal. I think this is, I guess 25 years in, might take another 25 to get there, but I do think that there is the possibility of pulling this off for organisms like the mouse. And we now have an initiative that's been co-funded by the Allen and the Chan Zuckerberg Institute to do this both at UW and the Allen as a C-Hub for synthetic biology. We're really trying to push this vision of essentially kind of turning a mouse into an iPhone is one metaphor for it here, or an Android or something. It should be an Android. It won't be locked down. So, maybe end on this slide, which is I think we often think about cell type atlases, and that's kind of become the metaphor that we use for where the intersection of developmental where genomics has been heading. But I think particularly as it intersects developmental biology, but I honestly think just more generally, right, omnicellular, ecellular, it would be better for us to think about framing this as a tree. And not a strict tree in the Selson sense, but more of like what I would term a consensus ontogeny, right? Where you think about the process that gives rise to a mouse, including aspects that are consistent as well as inconsistent from mouse to mouse, are both of interest, right? We want to know what always happens the same, but we also want to know what the variability is on that is somehow tolerated to give rise to essentially an identical phenotype. And I think, coming back to the theme of genetics, I think this will loop back to genetics, where, we will essentially, in my ideal future, these kinds of decorated trees, which are read out at a single endpoint, but give you a glimpse into both present but also past, allow you to compare the present and past of wild type, but also genetic variation, including mutants, and to be able to decipher what went wrong and precisely what went wrong and when it went wrong and where it went wrong. And more generally, the other piece here, I think, as we think about even amongst wild type animals, how to actually piece all this information, I can imagine a future in which we, oh man, I have a typo there. That should say mammalian, that should say developmental genetics or developmental biology. I do think the field is going to move towards, or I hope the field will move towards more of a statistical framing for how we think about development unfolding, which is quite different from the Selston model, but I think will be useful as we think about phenomena like variable expressivity, incomplete penetrance, and just more generally how it all happens, right? And thank you all for, oh no, there we go. Okay, thank you all for listening, and tons of acknowledgments, some of which I did along the way, but really this has been a massive effort between our lab and collaborators over some years now, and this is all thanks to them, and I'm just the messenger here. So thanks for listening. Question slide. Okay. Yeah, Kyle.
I have a question about the gastruloid heterogeneity and when the transition from naive to time split potency occurs, whether that explains the outcome that you see. If we just go to some neutral media, the mix of naive cells and time cells, when both of them mature, they both react very, very differently.
Yeah, and that's a great point. It's not something that we've teased out yet. I mean, I imagine when we go to a monoclone, like a monoclonal founder, maybe we're picking from that distribution in a sense.
Right. Yeah. So maybe before the naive or time states, then you could look at it under live, like have a couple of live imaging.
Yeah. Or are there media conditions that would allow us to, yeah.
Go to the dry and lit together all the cells, naive state, without increase or decrease.
Yep. Yep. I will say that last experiment where we did the founder of founders was the least heterogeneity we'd ever seen. And I think it's in partly because we're making the founders so related to one another in a sense, right? And so that would be consistent with your hypothesis. Oh, I don't know. Go ahead.
Thank you for the cool talk. I was wondering for the lineage tracing experiments where you identify like at what lineage you start seeing the bifurcations, like how do you determine the lineage? Because the prime editing like systems isn't, where do you kind of get that information of when the
even when I'm saying it's the first division, the second division. Yeah. So these trees go back to a single, they're monophyletic, right? So we can see a number of prime editing events that occurred before the first cell division and additional events that occurred between the first cell division and the second cell division.
Isn't like prime editing events not correlated to, how is it correlated to cell division?
That is a great question that we're exploring. It's more about like, I don't think we can correlate it with absolute time, but because we have because we have robust sampling of each branch of that first cell division, you can tell it's the first cell division, right? Unless all the daughters from some other cell division died, in which case we couldn't. But barring that, right, you can see it. So, yeah.
So for the recording, the typewriter, so what is the theoretical upper bound? Some of the events are pretty fast.
Also a great question. So it's still a little unclear how quickly prime editing happens, right? Or how quickly it can happen, right? Or especially the NGRAM activation, those steps have to happen. This is not going to be on the resolution of seconds or minutes or even 1 hour, right? I think it's going to be on the resolution of multiple hours, we have some preliminary data that puts us well under a day. So like, if I had to guess right now without being too firm about it, I'd say 6 hours is probably going to be about where we're at, right? For any given tape, right? So, but across a multitude of tapes, you might get higher resolution than that because they're not going to all be synchronized, but there will be some lag there. So even if we're eventually able to record, imagine we could record neuronal activity at some level. If we could, it still is going to be a very different kind of thing we're measuring than what you get with patch clamp. We'll be looking at activity over days, weeks, months, right? A higher resolution. I'm happy to stick with this problem until we solve it. So, yeah. Oh.
Yeah, is there a way to tune the dynamic rate of editing for things on that scale at the same time, working on slower time? For some of the things we're talking about, like metabolism, like stuff like that, days and weeks, like the over hours.
Yeah. So I guess there's kind of two questions. I thought your question was going in a different direction when I went in a different direction. So there's a cyclic aspect. I guess there's one aspect, which is how do you correlate things with absolute time? That's kind of one question, right? Where's your question starting?
Yeah, sure. Is there a way to get absolute time?
So there's time stamping, right? So imagine you had docs and something that was like docs, like, I don't know, some other small molecule. You could imagine alternating, right? And essentially getting timestamps across your tree that could be aligned and essentially working in absolute time that way. I think that's entirely possible. I think it's easy, slowing down editing is probably easier than speeding it up, right? I think it'd probably take a more detailed understanding of exactly what's rate limiting in prime editing to kind of speed it up, or adaptation of the concepts here to other kinds of editing that are symbolic and can operate on faster timescales, which is another possibility. Right? Yeah.
That could be a heterogeneity question. Would you have a sense of the relative contributions to the heterogeneity of, you highlighted the fantastic effects of what happens if the pathways get activated. So also is presumably some degree of somatic error actually happening with the MBLA set
You mean karyotype?
Yeah, the reason there was total heterogeneity at the beginning, having karyotype went forward to make
we could, it's relatively easy to pick out large scale karyotypic abnormalities from single cell RNA-seq data, especially if you're aggregating at the level of clades, which we have. We have not done that analysis, but we probably could and should. So that would answer that one, right? I do think the spatial piece is playing a component here, but I think it's somehow linked with the more stochastic piece, and they're somehow interplaying, so to speak. I think that's my baseline prediction. Whether there's something else, yeah. If not, and there's a reason for it. So I think in general, like one of the most, I guess there's a few reasons for it. One, C. elegans develops incredibly quickly, right? And plus we already know the answer, right? So that, but the time span in which it occurs, which it happens, means that you wouldn't necessarily be able to get robust recording across that time frame. Prime editing I don't think is super well fleshed out in C. elegans to my knowledge, or even with the Gestalt type methods. I think the other concern there was that in general these kinds of methods require or benefit from the single cell methods that we use are often lossy, and if you're trying to profile one organism, one animal, and you're only recovering 10% of cells, that is something that a mouse will tolerate and still give you useful insights. But if you're doing it in C. elegans, it will not go so well. So those are some of the reasons. Yeah.
What are your ideas?
Yeah, it's a great question. I think the canonical theories about, like, to the extent that epidemiology has been done in these long-term outcomes for kids born by vaginal versus elective C-section, right? I mean, the data is kind of, I don't think it's like completely solid, but to the extent it's been done, I think the explanations have centered around the microbiome and something around the passage through the canal and the mechanical forces then having some impact, right? I don't think we've ruled either, none of this was germ-free or anything like that. We haven't ruled any of that out. I think because Ian went to lunch, I don't know how controlled we are for things like whether the mice were, unequivocally, like whether there's any nursing happening or anything like that. So I think basically we need to do, and we are now in the process of doing, much more careful experiments where we're really tracking all the metadata and trying to, nail down what factors contribute. My guess is it's probably all of these things are contributing in different ways. But yeah. So I would say that